Today we will talk about the dihybrid cross. What do you mean by dihybrid cross? This dihybrid cross, it deals with the inheritance of two different characters. That means here we study two different characters, and this is what is called as dihybrid cross. Di means two. That means we study inheritance of two different characters, or what we say that the inheritance of two different traits. Or we know that one character, one trait, it is controlled by one gene, and one gene has got two pairs of alleles. One gene has got one pair of alleles on the homologous chromosome. Obviously two characters will be controlled by two genes, and two genes have got two pair of alleles. So we can say that this dihybrid cross, it considers the inheritance of two genes or two pair of alleles. So it considers the inheritance, or it considers the inheritance of two genes, two different genes, or two pair of alleles. Two pair of alleles. So what do we mean by dihybrid cross? A dihybrid cross is a genetic cross where we consider the inheritance of two different characters, or we consider the inheritance of two different traits, and one trait is controlled by one gene. Therefore two traits would be controlled by two genes. So we can also say that this dihybrid cross is that cross which considers the inheritance of two genes or two pair of alleles. Then what is the inference of this dihybrid cross? What is the result of this dihybrid cross? It says that inheritance of one character, inheritance of one gene, is independent of the inheritance of the another gene or another character. That means one character, it will transmit from one generation to another generation in one way, and while as this character, it doesn't have any sort of relation with the other character, which will transfer from one generation to another generation in the living organism. So we can say that what is the inference? What is the inference of dihybrid cross? That means what is the outcome? What this dihybrid cross wants to say? It says that the inheritance of one character, the inheritance of one gene, is independent of the inheritance of the another gene or another character. That means inheritance. That means the transmission of character from one organism to another organism. Inheritance of one trait. Inheritance of one trait or one gene, we can say. Inheritance of one trait or one gene is independent of the inheritance of another trait, another gene. Inheritance of one trait, one gene, is independent of the inheritance of the another gene or another trait during their transmission from one generation to another generation, or when they get transmitted from parents to offsprings. They don't have any sort of association with each other. One gene goes independently without any sort of association with the another gene from one living organism to another, from parents to offsprings. So this is the inference of the dihybrid cross. What is the I will show you the what is the dihybrid cross. In case of a dihybrid cross, what is the phenotypic ratio? What is phenotypic ratio in case of dihybrid cross? phenotypic ratio in case of dihybrid cross is 9:3:3:1. This is the typical phenotypic ratio in case of dihybrid cross. And what is the genotypic ratio in case of dihybrid cross, which can be asked in the examination? Genotypic ratio in case of dihybrid cross, it is 1:2:1:2:4:2 1:2:1. So phenotypic ratio in case of dihybrid cross, phenotypic ratio is 9:3:3:1, and genotypic ratio in case of dihybrid cross is 1:2:1, 1:2:1, 2:4:2, 1:2:1. This we can also calculate this phenotypic ratio and genotypic ratio in case of dihybrid cross. We can also calculate it from the this monohybrid cross. So see, we have this monohybrid cross. How we can calculate it from the monohybrid cross? In case of monohybrid cross, phenotypic ratio in case of monohybrid cross. We can calculate this dihybrid phenotypic ratio from the monohybrid cross. That means monohybrid phenotypic ratio. In case of monohybrid cross, we have phenotypic ratio that is 3:1. So 3:1. So it would be like this: 3:1 into 3:1. Same is the case. We can calculate this genotypic ratio in case of dihybrid cross from the genotypic ratio of monohybrid cross. We know that genotypic ratio in case of monohybrid cross is 1:2:1. It is easy to remember, 1:2:1. So in case of dihybrid cross, it would be like this: 1:2:1. So we can calculate it like this. We have 3 3s are 9, 3 1s are 3, 1 3s are 3, 1 1s are 1. So 3 3s are 9, this 3 1s are 3, this 1 3s are 3, 1 1s are 1. So this is the typical phenotypic ratio in case of dihybrid cross, which we have calculated from the monohybrid cross. Same is the case. We can also calculate this genotypic ratio in case of dihybrid cross from the monohybrid cross genotypic ratio. So we have 1:2:1, 1:2:1. As we are taking two characteristics, so we have to take the multiply of this genotypic ratio in case of monohybrid cross. So it would be this: 1 1s are 1, 1 2s are 2, 1 1s are 1. Same is the case: 2 1s are 2, 2 2s are 4, 2 1s are 2. So 1:2:1. So 1 1s are 1, 1 2s are 2, and 1 1s are 1. So we have got this is the typical genotypic ratio in case of a dihybrid cross. So the phenotypic ratio in case of a dihybrid cross is we have phenotypic ratio in case of dihybrid cross that is 9:3:1, and genotypic ratio in case of dihybrid cross we have 1:2:1, 2:4:2, 1:2:2. We have calculated it from the monohybrid cross. That is why we have phenotypic ratio 9:3, phenotypic ratio 3:1, and genotypic ratio 1:2:1. So how this cross was performed? We will explain this cross. Explanation or illustration. How will you explain this cross? I told you that dihybrid cross is that cross which deals with the it deals with the inheritance. Dihybrid cross is that cross which deals with the inheritance of it deals with the inheritance of two different characteristics or two different traits. So for dihybrid cross we need two traits, two characters. One is, for example, we'll take seed shape and seed color. Seed shape and seed color. To explain the dihybrid cross, it is necessary that two characters will be taken into consideration. So we have taken two characteristics. One is seed shape, another is seed color. That means shape of seeds and shape of seeds and color of seeds. Shape of seeds and color of seeds. So we have two shapes for the seeds. One is dominant, another is recessive. That is round seeds and wrinkled seeds. In pea plants, in some varieties of pea plants, seeds are round, and in other varieties of pea plants, seeds are wrinkled. This round is a dominant trait and wrinkled is a recessive trait. Same is the case, we have seed color. In some pea varieties, we find seed color is yellow color seed. And in other varieties of pea plants, the seed color is green color seeds. Green color seeds. This yellow color, it is dominant over the green color. That we have explained in the monohybrid cross. Now we'll take Mendel crossed two pea plants. We'll take parents. We'll take phenotype of the parents. It is pea plant with round and yellow seeds. It was crossed with the pea plant with wrinkled and green seeds. Phenotype is pea plant with round and yellow seeds. It is crossed with pea plant with wrinkled and green seeds. Wrinkled and green seeds. Wrinkled and green seeds. So obviously these are we have to, for example, these are two pea plants. This is one pea plant, this is another pea plant. In this pea plant, the seeds are round and the color of seeds is yellow. In another pea plant, the seeds are wrinkled and the color of seeds is green. So then these two pea plants were crossed. Cross means that it is simply a sort of matting, where in case of plants we take the pollen grains from this plant and it would be dropped on this plant. That is what is called as cross. Cross is sort of deliberate hybridization between two genetically different individuals so as to get the F1 hybrid, F1 offspring. So we have got this is pea plant with round and yellow seeds, pea plant with wrinkled and green seeds. Obviously, what would be their genotype? Genotype is simply we have pea plant with round and yellow seeds, the dominant. Round trait, it is a dominant trait. So capital R, and yellow trait, it is a dominant trait. It would be like this, and this condition is pure, homozygous. That means capital R, this is pure or homozygous for the trait. Same is the case, capital Y, it is a pure or homozygous for the trait. We will take here we have pea plant with wrinkled and green seeds. Wrinkled is the recessive trait, so we will write down small r, and green is the recessive trait of yellow seeds. small y, small y. So this is the genotype of these two pea plants. Then we'll make gametes. Gamete formation. Gamete formation, it follows the law of segregation. That means it follows the law of segregation. That means the gametes, during gamete formation, the alleles separate. One allele goes to one gamete, another allele goes to another gamete. So it would be like this. We have capital R. This is, these two alleles will separate. These are the two alleles of a gene, they will separate. These are two alleles of a gene, they will separate. This, we have one gamete, genetic constitution is capital R, capital Y. Another gamete, genetic constitution is this. So these are two gametes which are formed. Same is the case here. Small r, two alleles of a gene will separate. This is two alleles, they will separate. Two alleles, they will separate. Gametes have only one allele for the gene, so gametes are always pure. So this, then we will make random fertilization to get the F1 generation. This is fertilization, what is called as random fertilization. That means the gametes will fuse with each other, and which gamete will fuse to which gamete, it is a matter of chance. So we have this combination in the individual, capital R, small r, capital Y, small y. So this is what, this is F1, first filial generation, or we can say that F1 generation. This is first filial generation or F1 generation. And obviously what, this is the genotype, this is the genotype. What is the phenotype? This is a pea plant with round and yellow seeds. Yellow seeds. Although it is whether it is homozygous or heterozygous, it is obviously, it is hybrid. It is because it is formed by the cross between two genetically different parents. This is one parent, this is another parent, genetically different parent. So we get the, this is heterozygous. Heterozygous individual. Why it is heterozygous? Because we have, if we take these two alleles, they are dissimilar. If we take these two alleles, these are dissimilar. Heterozygous means that if you have got two dissimilar alleles for a gene, that condition is called as heterozygous. F1 generation, first filial generation. Same is the case, we can also get here. We can also get the same result here. This is capital R. This one, this is crossed here. Small r, capital Y, small y. Then this was first filial generation. Now, what Mendel did, he made selfing. That means F1 individuals were self-crossed with each other. F1 individuals were crossed with each other. It is a sort of inbreeding. That means the F1 individuals were crossed with each other, or self-crossed, we can say that. A sort of inbreeding where genetically related individuals were mated with each other. So selfing, that means these are F1 individuals. That means F1 individuals were crossed with each other. F1 individuals were crossed with each other. So F1 individuals were crossed with each other. When these F1 individuals were crossed with each other, obviously the seeds were collected, they were sown in the fields, and ultimately the F2 generation was raised from this cross. See what will happen. So this is our F1 generation. Now we'll make a gametes. gamete formation, capital R small r, capital Y small y. That means first selfing or this F1 generation, they will be crossed with each other. That means F1 individuals were crossed with each other. It is a sort of inbreeding, we can say that. We got F1 individuals, then these F1 individuals were selfed. That means they were crossed with each other. So these F1 individuals were crossed with each other. Then what we have to make? Gametes. Gametes. How many gametes would be formed? This is capital R, capital Y, capital R, capital R, capital Y. One gamete. This is capital R small y. Another gamete. Small r, capital Y, small r, small y. So four different types of gametes will be formed here. Same is the case with here. We have this capital R and capital Y. This is one gamete. Capital R small y, this is another gamete. Same is the case, small r, this is another. It would have combination with this allele, capital R, small r, capital Y. Then small r, small y. Four types of gametes are formed. Then there is now we'll make random fertilization. We'll explain it with the help of a checkerboard. We'll explain it with the Punnett square. It would be convenient to explain with the help of Punnett square. So we have, this is male gametes, female gametes, and what we'll get here F2. We'll get F2 generation here. We'll get F2 generation here. So we have gametes, four gametes, 1, 2, 3, 4. This is capital R, capital Y, capital R, small y. This is small r, capital Y, small r, small y. Same, these are male gametes. Then there is a female gamete. Obviously, this is here capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. They'll make a cross. So you can take as, this is simply as a male, this is you take as a female. Same is the case, you take here, this is male, you take here, this is female. Now we'll make the cross. Gametes are here, gametes are formed here. Then we explain it with the help of a Punnett square. So make a cross. We have got capital R, capital R, capital R, capital R, capital Y, capital Y. This plant would be round and yellow. Same is the case, round and yellow seeded pea plant. This is cross between this and this. We have capital R, capital R, capital Y, small y. This is the pea plant with round, and this is round and yellow seeded pea plant. Same is the case, we have capital R, small r, capital R, small r, capital Y, This is pea plant with round and yellow seeded pea plant. Same is the case, we have capital R, small r, capital Y, small y. So this is the pea plant with round and yellow seeded pea plant. Then we have got another, this capital R, capital R, capital Y, small y. This is also a pea plant with round and yellow seeds. This is the another, we will take capital R, small y, small y. This is the pea plant with round small y, small y. This small y, small y. This small y and small y. It explains what? Small y, it explains green. So round and green seeded pea plant. Small y, it explains green seeds. So this is a pea plant with round and green seeds. Then we have got capital R, small r, capital Y, small y. This is the pea plant with round and yellow seeds. Then we have another capital R, small r. This capital R, small r, small y, small y, small y, small y. This is a pea plant with round and small y. It is for yellow color seeds. Sorry, and this is for green. This round and green seeded pea plant with round and green seeds. So this small capital R, small r, capital R, small or small y. This is pea plant with round and green seeds. Then we have got here this capital R, small r, capital Y. This is pea plant with round and yellow seeds. So we have got capital R, small r. Then we have got capital Y, small y. This is pea plant with round and yellow seeds. Because capital R, it designates round character. Capital Y, it designates yellow character. Then we have got the small r, and capital Y. This small r designates what? This small r, this designates a wrinkled character, wrinkled. So this is a pea plant with wrinkled. This is the pea plant with wrinkled and yellow seeds. So small r and small r, this is wrinkled seeds. This is the genotype for wrinkled seeds, and capital Y, this is the genotype for yellow seeds. Then we have got this small r, capital Y, small y. So this is also small r. This is a pea plant with wrinkled and capital Y, small y, wrinkled and yellow seeds. Then another one, capital R and small r, capital Y, small y. The pea plant with round and yellow seeds. Pea plant with round and yellow seeds. Then we have got capital R, small r. This small y. Capital R. This is small r, small y. This is small y. So this is pea plant with round and small y. It is for the green seeds. It is for the green color. So we can say that small y, it is for the green seeds. So here we will have pea plant with round and green seeds. Green seeds. Then we have another one, small r. This is small r. Then this is also small r. This is capital Y, small y, capital Y, small y. This is the genotype of the pea plant. What would be the phenotype? Phenotype, it is small r, small. It is small r. In small r, it is for the wrinkled seed. So we can say that pea plant with wrinkled, and capital or small, pea plant with wrinkled and yellow seeds. It is a pea plant with wrinkled and yellow seeds. Then we have got small r, small r. This is small r, small r. This is small y, small y, small y, small y. This is the pea plant with small or small r. This is for wrinkled seeds, and small y, it is for the green seeds. So this pea plant would be wrinkled, pea plant with wrinkled and green seeds. So this was F2 generation. This was F2 generation. This was F2 generation. So this is what is called as F2 generation. F2 generation. Second failure generation. So this from this cross, what we got when we crossed F1 individuals, we got F2 generation. Second failure generation. Now we'll calculate the genotype for this. In the F2 generation, what we obtained? We obtained round and yellow seeds. Round and yellow seeded pea plants. We obtained round and green seeded pea plants. Green seeded pea plants. We obtained, then in the F2 generation we obtained wrinkled and yellow seeded pea plants. Yellow seeded pea plants. Then we obtained wrinkled and green seeded pea plants. How many total of one? We have 4, 6. We have 16 individuals. We have 4, 8, 12 and 16. Total we have got how many individuals? We have got 16 individuals. So it would be like this. Out of 16, how many are round and yellow? We have got 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6, 7, 8 and 9. We have got 9 round and yellow seeded pea plants. Out of 16, how many are round and green? Round and green. This is 1. Round and green. 1, 2 and 3. This is 1, 2 and 3. Round and green seeded. How wrinkled and yellow? This is wrinkled yellow. 1, 2, 3. 1 wrinkled and yellow. Second ring. Third. We have out of 16 we have 3 wrinkled and yellow. And how many out of 16? How many are wrinkled and green? Out of 16 we have 1 wrinkled and green. This is 1. This one. So the ratio is 9:3:3:1. So we have got 9, 3:3:1. This is a typical phenotypic ratio in case of dihybrid cross. Whereas genotypic ratio is simply genotypic ratio is simply 1:2:1:2:4:2:1:2:1. That means what is the genotype of these individuals? How many? This indicates how many. Genotypic ratio gives us idea how many are pure hybrids, or how many are heterozygous, how many are homozygous individuals. So this dihybrid cross, it was used by Mendel to deduce what? It was used by Mendel to deduce the law of independent assortment. Law of independent assortment. That means from this dihybrid cross, the Mendel deduced law of independent assortment. That we'll discuss later on. What do you mean by law of independent assortment? It says that inheritance of one character is independent of the inheritance of the another character, or inheritance of one gene is independent of the another gene during their transmission from parents to offspring, so from one generation to another generation. That is law of independent assortment. We can also, this inheritance of two characters, it follows the what? It follows Predictor Rule. Predictor Rule. When we take the inheritance of two characters, we have taken two characteristics, seed shape, seed color. This the inheritance of two characters, it follows the Predictor Rule. Yeah, Predictor law of probabilities, one of the important rule in genetics. This is Predictor Rule, or Predict law of probabilities. Predictor law of probabilities. What it says that? It says that if this predictor law of probability says that if there are two events, for example event A and B, then the probability of these two events to occur simultaneously is equal to the product of their individual probabilities. Or we can say that what is the predictor law of probability? The probability of two events occurring simultaneously. Probability of two events occurring simultaneously can be calculated from the product of two events occurring simultaneously can be calculated from the probability of their product. If we have two events, probability of two events occurring simultaneously can be calculated from the product of the individual probabilities. That means if you have got two events, for example, this is you have got two events, event first and event second, event A or event B, they are occurring independently. The product of these two events A and B to occur simultaneously can be calculated by the product of their individual probabilities, that is equal to the product of their individual probabilities. It can be explained like this. We have individual probabilities. For example, we have individual probabilities. Individual probability, for example, we have round seed, we have round seed, we have yellow seed, we have wrinkled seed, we have green seed. What is their individual probabilities? That means individual probabilities, that is 3 by 4. This is also 3 by 4. This is 1 by 4. This is 1 by 4. That means how you can calculate the probability? Probability can be calculated by total number of events. Total number of events. This is number of favorable events. Number of favorable events. So probability of an event. So in case of this round series, we have this probability, yellow series have this probability, wrinkled series this probability. Now what are their combined probabilities? Combined probabilities. Combined probability can be calculated by taking the product of their individual probabilities. By taking the product of their individual probabilities, we have a round and yellow seeds. We have a round and yellow. We'll take their individual probabilities. We have round and green seeds. What is the probability? 3 by 4. This is round, 3 by 4. What is green? 1 by 4. This is 1 by 4. Round. Then we have wrinkled and yellow seeds. What is for the wrinkled? Wrinkled for 1 by 4. What is yellow? It is 3 by 4. Individual probability for yellow seed 3 by 4. Then we have wrinkled and green seeds. Wrinkled and green seeds. What is the probability for wrinkled seeds? 1 by 4. What is the probability for green seeds? This is green seed, 1 by 4. Now we can calculate their combined probability is equal to the product of their individual probability. So we have 3 3s are 9 by 4 4s are 16. 3 1s are 3, 4 4s are 16. 3 1s are 3, 4 4s are 16. 1 1s are 1, 4 4s are 16. So that is 9:3:3:1. So we have calculated. It means that the two inheritance of two characteristics, it follows the product law or product rule or probability law or product law of probabilities. Product law of probabilities. Hope you have understood it. So if two events, the probability of two events occurring independently is equal to the product of their combined probabilities. Probability of two events occurring simultaneously can be calculated from the product of the individual probabilities. This is called as Predictor Rule or Product Law of Probabilities. Thank you so much.